Good morning, everyone. I want to officially welcome you to today's webinar, Fungicide Rotation Program Development and Implementation, presented by PBI-Gordon and taught by Koch, Ph.D. My name's Lisa Wick. I'm the Senior Manager of E-Learning Programs here at GCSAA and will serve as today's moderator. We are recording today's event, and you will have access to that recording as part of our follow-up email. Your audio is muted in this system, but if you want to ask a question, we welcome you to do so. You can do that by clicking the raise hand icon on your GoToWebinar control tab, and we can unmute you, or you can type into the question answer box, and we will take questions as we go along. The GoToWebinar control panel also has several handouts in it, the slide set and some others that Paul has provided. If your control panel has minimized, you'll see a little narrow strip on your screen at the top of which is an orange rectangle with an arrow in it. If you click on that, that will open that control panel back up where you can access those handouts or type in questions. Today's session is eligible for 0.1 GCSA education points, and at the end of the program, we'll give you the code that you can put in the association's website to get those points. We hope that you find today's webinar valuable. It is brought to us in partnership with PBI-Gordon. With a full line of herbicides, insecticides, fungicides, growth regulators, and other products, PBI-Gordon Corporation is a national leader in the professional turf and ornamental management industry. From proven favorites like the Trimec line of broadleaf herbicides to innovative products like new Tekken broad-spectrum fungicide, PBI-Gordon delivers performance and service that turf pros have counted on for over 70 years. Today's presenter, Paul Koch, Ph.D., is an assistant professor in the Department of Plant Pathology at the University of Wisconsin-Madison. Koch's research focuses primarily on developing precision disease management strategies for snow mold and dollar spot in turf grasses and investigating the fate and impact of turfgrass pesticides in the environment. Koch also oversees the University of Wisconsin's Turf Diagnostic Lab and the UW Turfgrass Fungicide Testing Program. Please join me in welcoming today's presenter, Paul Koch. Paul?
All right, thank you for the introduction, Lisa. Thanks to PBI-Gordon for sponsoring the session today and GCSAA for the invite to come and talk to you about fungicide rotation program development and implementation. Happy first day of spring to everybody. I know for many of us it doesn't feel like spring. High of 37 here in Madison today. Snow forecasted for the weekend. The Northeast is getting a ton of snow again this weekend. Before you know it, though, we'll be anxiously awaiting that first day of fall, so that summer goes by pretty quick. So let's go ahead and get started. I'm going to turn off my camera to avoid, some of the glare that you're seeing from the top of my head throughout the session today. And as Lisa talked about with the questions, if you have any questions, please go ahead and feel free to ask any questions throughout. I'll be checking with Lisa a few times throughout the session today to see if anybody has any questions. So why don't we go ahead and get started and see if we can get the slide to...
Because I messed with the mouse, you have to click the screen again.
Okay, I will click the screen, and there we go. Let's go ahead and talk about our current situation. Where do we find ourselves in with regards to fungicides, the availability of fungicides? In my mind, it's likely that we're going to have fewer fungicide options available to us in the future, especially when we're talking about different fungicide chemistries. There's a lot of new chemicals coming out of the market in the next couple of years. Many of those are going to be from the succinate dehydrogenase inhibitor class of fungicides, the SDHI class of fungicides. With that addition to the market, an important market, especially when we're talking about dollar spot control, but with that addition, we really have three major classes of fungicides. We have our DMI class of fungicides, we have our strobilurin or our QOI class of fungicides, and then we have our SDHI class of fungicides. We mix in a couple other important products like chlorothalonil, obviously an important piece in resistance management, clozidam or Secure, Iprodiol. These are other important products, but as classes, we really have three primary classes that we use. This relative lack of diversity in our fungicide classes puts higher resistance pressure on those existing chemistries. So not only do we have fewer new chemistries coming to the market, we have a higher pressure for resistance because we don't have a lot of rotational partners. In addition, if we look to the future, we see potential restrictions on some of our really important resistance management products, most notably chlorothalonil. Now, there's no proposal from the EPA to further restrict or eliminate chlorothalonil from the market, but if we look at our friends to the north in Canada, certainly there are some significant restrictions on chlorothalonil that have been imposed there. I don't know if they ever fully went into effect or fully been approved, but I know when I was up there last fall, there was a proposal on the table to limit the number of chlorothalonil applications to 1 or maybe 2 throughout the season. So that means future resistance management strategies will be increasing in importance. So I think it's important to have a good idea of what is important with regards to developing effective resistance management strategies. So before we get any further into the talk, let's go ahead and learn a little bit about you guys. Where is your course located? Do we have some different locations from around the U.S.? And then if you're outside of the U.S., please go ahead and type your country into the Q&A box. Obviously, with different diseases that are prominent around the country, we have different primary disease programs. We'll talk a little bit about those programs near the end of the presentation today, but knowing where everybody is located will give me an idea on the major diseases that we're concerned about as far as resistance development.
Okay, closing the poll in 5, 4, 3. Do you want to guess at all what segment is?
I'm going to guess Northeast and Midwest are pretty close to being number 1. Probably nearly tied is my guess.
Oh, man.
Oh, okay. Midwest. Top. Way to go, Midwest. Way to represent. Okay. So yeah, what this tells me then is when we're talking about resistance in this particular session with 45%, nearly half of the participants from the Midwest, we're primarily focused on dollar spot. And there's a lot of information, a lot of research over the decades that has been done on resistance in dollar spot. Obviously, dollar spot resistance is important in the Northeast as well. Other diseases primarily important in the Pacific Northwest, in the Southeast. And we'll touch on some of those as we move throughout the session today. All right, brief outline on what we're going to talk about. Certainly, we're going to talk about fungicide resistance. But before we get to fungicide resistance, I really want to focus on more how do fungicides interact with fungi in the environment? How do fungicides work on fungi? We need sort of this basic introduction to how fungicides work and how they interact with fungi to really determine and gain a better understanding on how fungicide resistance develops. So after we have an understanding of how fungicide works, that will then lead into a discussion about how fungicide resistance develops. We'll spend a few minutes on how fungicide resistance develops in turf fungi, and then we'll spend the last portion of the talk talking about strategies for mitigating or slowing resistance development in your program and at your facility. Notice I did not say strategies for eliminating or stopping resistance. It is my opinion that it is not a worthwhile goal to completely eliminate resistance. I think as long as we use fungicides, we're going to have some resistance develop over time. The key then is slowing that resistance as much as we possibly can to maintain the integrity or the effectiveness of those products, those fungicides, for the longest duration possible. All right, so before we go any further, though, of course, we need to talk about the disease triangle. I am in the Department of Plant Pathology at Wisconsin, as Lisa said, so I'm contractually obligated to show a disease triangle picture at any talk that I give, and many of you are familiar with this. We won't spend much time on the disease triangle. What the disease triangle states is that for a disease to occur on any plant, not just turf, we need a susceptible host, we need a virulent pathogen, and we need a suitable or optimal environment for that pathogen to infect that host. When we're talking about susceptible hosts in turfgrass, clearly there's variability in the level of disease susceptibility amongst different hosts, but for the most part, we consider most of our turf grasses as hosts for most turfgrass pathogens. For the purpose of today's talk, let's say that most of our turfgrass hosts are susceptible to various pathogens. Now, for our pathogens, we consider most of our pathogens to be ubiquitous out in the environment, meaning they're pretty much everywhere. So we consider dollar spot to be pretty much everywhere out on the golf course. We consider leaf spot to be pretty much everywhere out in the golf course. Now, this is not entirely accurate, right? There's certain diseases certainly that come and go, and rust is a really good example of one that is not always present out in the turf system. But for most of our primary diseases, we consider them basically everywhere out in the turf system. So really what we're waiting for then is the conducive environment. Most of our diseases need some level of moisture to cause disease. Not all, but most of them need moisture to cause disease. Some are when it's cool and wet, some are when it's hot and wet, but most of our diseases need some sort of moisture. All right, so let's get into talking about how fungicides interact with fungi. So this is a slide that I took from Dr. Rick Latton's book, A Practical Guide to Turfgrass Fungicides. This is an excellent book. I'm just going to go ahead, if anybody out there is interested in how fungicides interact with fungi in a turf-specific system, I highly recommend going out and buying Dr. Latton's book. You can buy it from the American Phytopathological Society website. I'm sure there's some other, I'm sure it's on Amazon somewhere. But if you want more information on turf fungicides, turf fungi, how the two interact with each other, this book is a great, great resource. It's really easy to read, got a lot of nice pictures. Rick did a really nice job on that book. So go out and buy that book. There's two primary things I want to focus on this slide, and that is that fungicides interact with fungi in different ways. If we kind of go clockwise, let's start with this microtubule over here in the upper right corner of the slide. Microtubules are simply parts of the fungus that pull the cell apart and are important in cell division. So thiophenymethyl and fluoropicolide are really important in inhibiting that cell division. Polyoxin D is really the only fungicide we have that inhibits cell wall development. If we go down here to mitochondria, mitochondria are the energy-producing areas of the cell, so anything that inhibits or disrupts mitochondrial activity is going to inhibit or disrupt energy production in the cell. So the QOIs, or the strobilurins, they interact and disrupt a certain part of the mitochondria. The SDHI fungicides localize and interact and inhibit a different part of the mitochondria. So QOIs and SDHIs act in very similar areas of the fungal cell, but they interact in different areas of the mitochondria. Mefenoxam, used for a lot of omycete or pythium control, is really the only one that gets inside the cell nucleus, interacts with the nucleic acid or the DNA. And we have many fungicides interact with the cell membrane. Right? So the demethylation inhibitor class of fungicides, DMIs, those are really the main ones that interact and inhibit cell membrane formation. If we have a problem with, if the fungus has a problem with cell membrane formation, it can't hold its contents inside the cell, it leaks, and eventually the cell dies. The last one here, really important one for resistance management, obviously, is chlorothalonil. Now, Dr. Latton here just has general cell constituents. What does that mean? Really, what chlorothalonil does is just, it mucks up the whole system. It binds very tightly to a very important molecule that does a lot of different activities in the cell. It's a molecule called glutathione. And when it binds up those glutathiones, it makes them unavailable to do the other process the fungus needs. And so, so the fungus eventually dies back. Now, what's important is that there's no mechanism the fungus has to get around this mode of activity of chlorothalonil. So it can't make more glutathione. It can't do other things to get around this. So this is why there's really been no documented cases of resistance to chlorothalonil in the 60 plus years that chlorothalonil has been around, or 50 some years. Came out in 1966. So there's been no documented cases of resistance to chlorothalonil, whereas there has been documented cases of resistance to basically every other one of these fungi. The second thing that I wanted to focus on this slide was, see how Dr. Latton here has sort of gaps in the end of the fungal hyphae here, the mycelium, right? And so this is where the fungus is growing. Fungal hyphae grow out from the tip. They don't grow from the back. Dr. Latton's example that he uses in presentations is they're like roads. When you build a new road, you don't build it from the back, you extend from the front. And so as this hyphae would continue to grow, it would continue to extend out from the front. And so what needs to happen is, as the fungal cell grows, it is constantly breaking down the cell wall at the tip and reassembling as the cell goes. So as the hyphae continues to grow out this way, the cell wall is going to extend out here, and then it's going to extend out here as it grows. What's important about that for our discussion today is that all fungicides need to be absorbed through the growing tip. Fungicides are not absorbed back here. They can't be absorbed through a fully complete cell wall. They need to be absorbed through this the growing part of the cell wall where there are gaps inside the mycelium. So that'll be important for some of our later discussions. All right, so again, as I just mentioned, the fungicide is absorbed by the growing point of the pathogen or the tip of the fungal hyphae. So again, another slide from Dr. Lattin's book. The fungicide is in the environment and it is absorbed through the growing tip. Again, not absorbed back here, only through the growing tip, and the fungicide concentration will decrease the further it is absorbed into the fungal mycelium. So it's certainly going to kill the first couple of fungal cells, but as it goes back into the mycelium, it's going to be less, it's going to be more and more diluted, and eventually it's going to reach a point where it does not inhibit or kill any fungal cell growth. So that's important for how fungicides interact and how fungicides control various fungi in the field. Fungicides do not eradicate fungi from our turfgrass systems, right? This is why you need to reapply fungicides on a regular basis. You apply a fungicide, it gets absorbed by whatever fungus. Let's just say this is dollar spot. It gets absorbed by dollar spot. The fungus dies back to a certain extent, and then once the fungicide is out of the system, it's dissipated, it's broken down in the turfgrass environment, then the fungus starts to grow again. Right? This is why, again, this is a common question that we get from people who are not familiar with how fungicides work, people who don't work in turf. So I get this question a lot from GMs or people from the general public. Why do you need to reapply fungicides? If you apply a product that kills the fungi, why do you need to reapply it? Well, we know the reason is that it doesn't completely kill and eliminate the fungus. It just knocks it back, and the fungus grows back over time. In my mind, then, the duration of a fungicide's efficacy is not related to its persistence in the field. It's related to how much it stunts back the fungus, right? So if we have emerald, which typically we say for most dollar spot cases will give about 28 days of control, it is not persistent in the field for 28 days. It's typically persistent in the field for about a week or so. And then it takes about another 3 weeks or so for the fungus to grow back to the point where it will cause symptoms. So there has been some research that supports the fungicide persistence aspect of this, of what I just discussed. Again, Rick Lattin, Purdue University, did a study looking at how long fungicides persist in creeping bentgrass fairways related to their control. And he worked with several different chemicals. Let's focus on azoxystrobin and metconazole. Azoxystrobin and QOY, we would expect to get 28 or more days of brown patch control from that product. Metconazole, a DMI, we'd probably expect maybe 14, maybe 21 days of brown patch control out of that product. So Dr. Lattin measured the concentration of both of those fungicides over time. And here with azoxystrobin, the bottom here is cut off a little bit, but we still get the gist. Azoxystrobin right here is about the 10-day mark on this slide. So there is basically no more azoxystrobin left in the system after about 10 days. But we still get brown patch control for 28 days. Let's look at metconazole. Again, here's the 10-day mark. After 10 days after application, we're down to about 0 fungicide left after 10 days. So even though both fungicides, both metconazole and azoxystrobin, are completely eliminated from the system within 10 days, they still provide control for a prolonged duration. We did some similar work at Wisconsin. We published this back in 2015, looking at influences of temperature on the persistence of chlorothalonil and iprodiol, so 2 very different chemicals than what Dr. Lattin looked at, but we found a pretty similar result. And so here, this is iprodiol on the left, this is chlorothalonil on the right. This top line is at a constant temperature of 10 degrees Celsius, or about 50 degrees Fahrenheit. This middle line with the white circles is iprodiol on bentgrass turf at fairway height at 20 degrees Celsius, which is about 70 degrees Fahrenheit. and then 30 degrees Celsius is about 90 degrees Fahrenheit. And right here is the 14-day mark. So what we found is that at normal summer growing conditions between 70 and 90 degrees Fahrenheit, within 14 days, that fungicide is gone from the system. So really agreeing with a lot of the results that Dr. Lattin showed. So how then does this relate to fungal disease development? So here's kind of a graph I made up. We don't really have data to support this at this point, but here on the left side of the graph, we have sort of a made-up term, fungal growth, and I have some numbers that correspond with fungal growth in the turf. I have assigned sort of a generic value of 25. Anytime the fungal population is over 25, we see symptoms. So for the purpose of this conversation, let's say we're talking about dollar spot. So anytime the dollar spot fungus gets above 25, we see symptom development. Now I have two different fungicides, two very different fungicides, emerald, boscalid, or daconil, chlorothalonil. Emerald, let's say we apply it at the 0.18 ounce rate. We expect to get 28 days of control out of that. Let's say we apply daconil at the 5.5 fluid ounces rate. We would expect to get 14 days out of control of that. Now we know from the previous couple slides I showed you, the fungicides persist in the environment for about the same amount of time. It's really the differential impact on the fungal population that leads to their different reapplication intervals. So if we kind of just advance the slide, let's say this is the fungal population in response to emerald. So here we have the fungal population is growing. Typically in southern Wisconsin, we would expect to see dollar spot symptoms occur somewhere in mid to late May. So we're going to make that first emerald application. The fungal population then will plummet back in response to coming into contact with that emerald. The emerald will stay around for about a week or 10 days in the system. There's going to be no growth by the fungus then. But as the emerald dissipates from the system, it's going to the fungus is going to slowly grow back over the next 3 or so weeks until it about reaches that 28-day interval where it's going to cause symptoms again. We make another application throughout the year. It comes, the population plummets. It slowly recovers. We make another application. It slowly recovers. Now, the impact of daconil then on that same system, as evidenced by this red line, we make the application about the same time in mid-May, but it is stunted back less than the emerald. So the daconil does not stunt back the dollar spot fungus as much as the emerald does, and so it comes back faster. The fungus recovers to the point where it can cause disease faster related to emerald. So again, it's not the fact that daconil is in the system shorter and that emerald is in the system longer. It's that they have different impacts on the dollar spot fungus. Right, so I don't have any data to support this. This is a theory of mine. I think it's a pretty solid theory, and we have received some funding from the United States Golf Association to look at this exact scenario, tracking the dollar spot fungus out in the field and how they respond to different types of fungicide applications. This is a study that we're on with Bruce Clark and Jim Murphy at Rutgers University and Gunhua Zheng at the University of Massachusetts as well. So,
Paul, when will that study begin? Are you starting that research now?
That research is starting this summer. So we're actually working right now on the development of the assay. We need to develop the assay to quantify and detect the fungus first. We're working on that right now. We hope to have that in place by the time we're starting to see dollar spot this summer. So we have that's a 3-year project. At the end of that 3 years, hopefully I have some actual data to support this graph I just made up and showed you guys. All right, so let's move then from sort of the generic how fungi interact with fungicides to how resistance develops. So there's really two primary types of resistance development in fungi. One is what we call receptor-mediated resistance, and this is the kind of resistance I think we're mostly familiar with. Fungicides need to bind at a specific site in the fungus to be active. If there is a mutation in that binding site, the fungicide will no longer be as active. They might not be active at all, or they might not be quite as active and just show minor resistance effects. It's kind of like a lock and key, right? So you have the key being the fungicide. If the key works in the lock, it opens the door. It can inhibit whatever process in the fungus that it was designed to inhibit. If the lock changes by mutation, then the key can't open the door. There's no impact on the fungus. We see resistance develop. The second way then is this non-receptor-mediated resistance. This is a newer area of resistance study that's really been going on really the last 5, 6, 7 years or so. And we'll talk more about non-receptor-mediated resistance in a few minutes. All right, so here another participant poll. Another participant poll here is, if you have observed resistance, how did you determine that? We always get questions from superintendents. So I guess the first question here then is, have you ever observed fungicide resistance at your course? So I guess we'll go ahead and let you answer to that one first. And then the next question is going to be, if you observed that you, or if you think you have observed fungicide resistance, why or how did you think that you had resistance? So Lisa, as they're answering questions, or as they're answering the poll, is there any questions in the forum?
We don't have any, but I have one for you.
Okay.
Because you mentioned in your study that you're working with Bruce Clark, so I would gather anthracnose is probably going to be one of the diseases that you're going to be looking at.
So oddly enough, no. As much work as Dr. Clark has done
He's done with anthracnose?
No, he's not done with anthracnose. No, by no means. But as much work as he has done in that, this is a dollar spot specific study. So we're not going to be looking at developing an assay to quantify anthracnose at this point. Maybe that's something. Maybe that's a follow-up study. But at this point in time, the goal of that study is just to focus on dollar spot.
Okay, and dollar spot is different from large patch, correct?
Correct. So large patch is a disease of warm season turf, and so not over half of the participants have not observed fungicide resistance. So that's a good thing, but it's still 38%. That is a pretty significant amount of people that have had that have observed fungicide or resistance. So then here's the follow-up question then. If you have observed resistance, for those 38% of you that have, how did you come up to that conclusion? Did you just didn't get any control? You had a breakthrough? Didn't get quite your full reapplication interval? You sent it into a lab? Somebody told you? All right, what are the results we have? So breakthrough near the end of the reapplication interval. And that's what I would have expected. That's the most common type of resistance that we see, is you expected to get 28 days of control from a product, and you got 24 or 22. Or if you wanted to get 21, you got 17 or something like that. We'll talk about the different scenarios where you might see absolutely no control from an application, but that is typically the most common, especially if we're talking about, dollar spot and DMI fungicides, or anthracnose and DMI fungicides, and some of those interactions are the most common that we see. I'm glad to see that several people have had their isolates tested by a diagnostic lab. There's not many of diagnostic labs that exist that can test for fungicide resistance. I know Dr. Jung at Massachusetts will test for resistance. We used to at Wisconsin, but it was just a pain to keep the reference isolates going, so we stopped that probably 7, 8 years ago. So that's a good tool to find a diagnostic lab that offers that service. All right, so let's go ahead and continue our discussion on how resistance develops. Now we're going to go back to a slide we've already seen. Right? We already saw this slide. This was from Dr. Lattin's book, one of the first slides that we looked at today. And depending on the fungicide and the site of activity, it can either be a single site, single gene mode of action, or it can be a site that is governed by multiple genes. So again, we'll start at the same spot, microtubules. 5-methyl, this is a classic single site, single gene mode of action. We just need one mutation in that fungus to confer complete and total resistance to a thiophantomethyl application. So you don't get a reduced control, you go from complete control to basically no control over a relatively short period of time. With the mitochondria inhibiting products, the strobilurins and the SDHIs, it depends a little bit on the fungus that you're talking about. So with SDHI fungicides and dollar spot, that appears to be a more gradual development of resistance that occurs in response to accumulation of multiple fungicides or multiple fungicide mutations. fungal mutations. The QOYs with anthracnose, that appears to be a more rapid development of resistance. Again, the DMIs with dollar spot, that is a slower development, gradual accumulation of multiple fungicides over time. And again, like we talked about before, with these gradual mutations, that typically manifests itself as a reduced duration of control. So you typically don't get no control. Typically what you see is a reduced duration of control. So looking more specifically then at, we can advance to the next slide. There we go. So talking more specifically, we went a little too far. Talking more specifically about the single site, single gene mode of action, thiophantomethyl. I'll move away the text box here. Here's a study we did looking at control of resistant dollar spot. Here's non-treated control, just sprayed water, and here's thiophantomethyl sprayed a few feet away. Again, we get almost no control. This is nothing against the Quality Pro thiophantomethyl. If we would have used 33, 36, or any other thiophantomethyl, we would have seen the same thing. Offers complete and permanent fungicide resistance. So only a single mutation is required. Relatively rapid development over resistance. If you're going to use thiophantomethyl on dollar spot, you would want to immediately include a resistance management program, and even then, it's still probably going to be not that long until you see the development of resistance. Again, the strobilurins and anthracnose have a similar, fairly rapid development of resistance. Moving on to then the DMI fungicides, or the DMI fungicides and the development of a more gradual quantitative level of resistance. This is sort of the foundational paper done in apples and an apple disease on DMI fungicides by Wolfram Kohler back in the mid-90s. And really what they showed is that the more DMI fungicides that an apple orchard was exposed to, the more resistant those populations were. So the white bars for both of these DMI fungicides are from orchards that had never been exposed to DMI fungicides, and you can see the level of resistance in this population is pretty low in both these. The stripe bars here are ones that received a moderate number of DMI fungicide applications over time. We see the population of this disease shift towards resistance, and then the solid black bars are the ones that received the most DMI fungicides over the course of the last, I think, the previous 5 or 8 years. And that is where the population is the most resistant. It continues to shift towards resistance in response to repeated DMI fungicide applications. So if we look at the initial population, these white bars, we do see that even without any exposure to DMI fungicides, there are some isolates that have some level of resistance. And so when we apply DMI fungicides, we're selecting for these resistant isolates then to grow and take over more of the population. On the flip side, when we have these more resistant populations, we do still have a small percentage of the population that does have sensitivity to the DMI fungicide. So again, multiple genes requires the accumulation of multiple mutations over time, and it leads to this gradual shift in the overall fungal population towards resistance, and that typically manifests itself as not reaching the full desired reapplication interval. Now let's talk about that non-receptor-mediated resistance. Again, we have two major types of non-receptor-mediated resistance. The first is that the fungicide molecule can be escorted out or transported out of the fungal cell prior to reaching that binding site. Remember, the fungicide needs to bind at that active site to kill the fungal cell. If it's removed from the cell prior to that, it will not be active. All fungal cells have these transporter molecules to some extent, but what's been found is that resistant isolates of dollar spot and other fungi have a higher percentage, have a higher number of these transporter molecules. On the flip side of things, or not on the flip side, but related, is the fungicide molecule can also be broken down or metabolized before it reaches the site of activity. All right, so these are done by what we call P450 oxidizing molecules. And again, if the fungicide molecule does not reach the site of activity in the fungus, it's not going to have any effect on the fungus. So again, resistant isolates have higher numbers of these metabolizing or breakdown molecules than sensitive isolates. Really, the only person doing work in this area in turf is Gunhua Zheng and his team out at UMass. And if you really want to get into some deep science, grab a couple of beers and download this paper of Gunhua's. And what he's shown here is that there are isolates, resistant isolates of dollar spot, that have higher concentrations or higher numbers of these transporter molecules that can shuttle the fungicide molecule out and confer resistance. So why is this non-receptor-mediated resistance important? Well, it allows more rapid development of resistance, and it also allows development of resistance to multiple classes of fungicides. If that transporter molecule can escort a DMI fungicide out, it can probably also escort out an SDHI fungicide out. And Goodwill has shown some of that in some of his work. All right, so for the last 10 to 15 minutes here, let's focus on developing resistance management strategies. I really have 5 strategies that I would list, and we'll go over each one of these in more detail. All right, so let's start out with maximizing plant health, right? And we're not going to spend really any time on this because this is you could spend an entire year of different seminars and webinars going over the various factors that lead to maximizing plant health. We're talking about things like proper fertility, proper mowing height, grass species selection, drainage, soil health. All that good stuff goes into maximizing plant health. The healthier the plant, the more likely that plant will be able to fend off fungal diseases or bacterial diseases or whatever it might be, and the less reliance on chemicals that is required. This picture was taken shortly after the U.S. Open this past year at Erin Hills. Zach Reineking and his team had the place in spectacular shape, really maximize plant health, and that's really an important part of managing for fungicide resistance development. All right, so we have another participant poll then. If you have observed resistance, what disease have you observed resistance on? Right, so I think the major resistant organisms that we typically see in turf are dollar spot, anthracnose, and pythium, but there can be others, depending on where you're located in the country. Lisa, any questions in the forum?
There aren't, but I have a question about this because you also do the diagnostic lab. How common is it that one of these fungal diseases is misidentified when something is sent to you? You know, that the superintendent thinks it's one thing, but then when you guys do the testing, that's not what it is.
Well, these 3 diseases listed here, dollar spot, anthracnose, and pythium, those are pretty easy to identify, especially dollar spot and anthracnose. But if we're talking about especially the root-infecting diseases, summer patch, take-all patch, those are really commonly misidentified just because we can see some fungus on the roots pretty commonly, but it's kind of depends on how much roots or how much fungus is on the roots and what are the other conditions. So I would say root-infecting fungi are fairly commonly misdiagnosed. I would throw pythium root rot in that as well, as far as a disease that's commonly misdiagnosed by the superintendent that we then identify at the diagnostic lab.
Because your attempt to remediate the disease is different, correct?
Yeah, I mean, certainly, especially with root-infecting diseases, a lot of times once you have the symptoms, it's not that easy to recover from those symptoms. So, but certainly getting an accurate identification of the disease, whether it's from a diagnostic lab or from another qualified individual, is really important. So, all right, not that surprising there. Dollar spot being far and away the most common disease that we've observed resistance to, and that's no surprise because it is the disease for many areas of the country that we spray the most for. So pythium, I imagine most people down there are probably from the Mid-Atlantic or Southeast that spray for pythium on a regular basis. All right, so when we're talking about choosing the right product, it's going to partly depend on where in the country we are. So let's wait for the map to show up here. There we go. So if you're located in the Midwestern part of the country and many parts of the Northeast and the central part of the country, really dollar spot's going to be your primary disease. You get plenty of other diseases, but you're going to build that program around dollar spot. I would argue in the Northeast and the Pacific Northwest, the primary disease you're building that program around is probably anthracnose. And in the Southeast, I talked with Bruce Martin and Jim Kearns down there. One of the major diseases, at least of bentgrass, is that pythium root rot. So these are the diseases that you would probably be most likely to see resistance occur because these are the diseases you're spraying the most fungicide for. So what are some resources that could help you choose the right product for the right disease? I would argue your local university extension specialist and their research is a great tool. We list all of our fungicide testing results on our diagnostic lab website. You can see the web address there. But based on where you're located in the country, locate your local extension specialist and get their opinion. A lot of industry representatives are really great resources on the right product for the right disease. I know some superintendents are leery that they're going to get a biased answer, but almost every rep that I know is if there's a better product than the one they sell that's going to work, they're going to probably make that recommendation because they know that if they give you a bad recommendation, they're going to see through that pretty quickly. So I think industry representatives, for the most part, are a great resource. And then other superintendents, especially with new products, see how it's working at their course and if it's going to be a fit at your course. One other really great resource then is the chemical control of turfgrass diseases. The last one was put out in 2017. This has been done for many years by Paul Vincelli at Kentucky. Bruce Clark from Rutgers came in to help with this past year. And really for almost any turfgrass disease, it lists the active ingredient and then lists its efficacy on a 1 to 4 scale, 4 being the most effective and then 3, 2, 1 being less effective. One thing I'm excited to announce is that we at Wisconsin have partnered with Rutgers in Kentucky to put their data, their fungicide efficacy data, into a searchable online database. We expect to have that ready by, hopefully, right around May 1. We're kind of in the final editing stages of that website, so I'm excited to hopefully launch that pretty soon. I think that'll be a great resource for choosing an effective disease control product. We're also going to have herbicide efficacy rankings on there as well, and we've partnered with Aaron Patton from Purdue on those rankings. All right, what about spraying only when needed? Why do we want to spray product only when we need to spray product? Well, one, there it makes sense, right? You don't want to spray any more product than you have to for financial reasons, but two, the fungus is only going to take up that fungicide when it's active. If we revisit the slide that we talked about earlier, right, there's only going to be those openings at the tips when the fungal hyphae is growing. So if the fungus is not growing and you put down a fungicide to control that fungus, it's a complete waste. It's not going to be taken up by the fungus at all. In addition, there's increasing evidence that the more fungicides that are applied, the higher the level of resistance, regardless of whether there's an effective fungicide resistance management plan or not. So here's some work that we did a while ago now. This was the mid-2000s when I was getting my master's degree. And we looked at thiophanate methyl and propiconazole sensitivity to dollar spot on golf courses in Wisconsin and Massachusetts. And what we found is that even within the same golf course, dollar spot sampled from within the same golf course had different levels of resistance depending on where they were sampled from. Obviously, we don't spray fungicides in the rough. Dollar spot populations from the rough had the lowest levels of resistance. We spray more in the fairways. Dollar spot isolates collected from the fairways had higher levels of resistance. Isolates collected from the putting greens had higher still levels of resistance. So if you are spraying needlessly, then you are increasing the possibility that resistance will occur. So what are some resources you can use to spray only when you need to? Indicator areas you can use. Those are marginally effective in my mind. They tell you when it's effective to spray at that area. It doesn't do you a real good job of telling where else in the golf course to spray. Environmental conditions, these can be soil temperatures, growing degree days. For dollar spot, we've just come out with a disease predictive model that I assume hopefully many people have heard of over the last couple of months. And here's the website if you want to learn more information about that model. It'll take you to a web page that describes the model, tells you all about the model, how you can use the model, offers you some tools to use the model, also provides links to Growing Degree Day Tracker and Greenkeeper app, which are 2 additional sites that you can use the dollar spot model through. All right, optimizing the fungicide application. Again, this is we won't spend any time on this because this is an entirely different seminar, but making sure your water volume is correct, you have the right nozzles in there, your spacing is correct. You need optimal coverage of the products over the turfgrass leaf blades to provide optimal control. A lot of times when a superintendent thinks they have fungicide resistance, they don't quite get that full 21 or 28 days of control. A lot of times the problem is related to this optimizing of the application. All right, so now let's talk a little bit about rotating and tank mixing product and product selection. So when you're doing this, the fungus has an impact. Dollar spot is what I would consider a high-risk disease for resistance to develop. Snow mold is not a high risk. It's a low risk. We're only spraying once or twice a year. It's a slow-growing fungus. We typically don't see much resistance development in snow mold. The product has an impact as well. We have high-risk products. Here's that thiophanate-methyl. It's a single-site, single-gene mode of action. Resistance is going to develop pretty quickly if we're using thiophanate-methyl on dollar spot. However, thiophanate-methyl used on snow mold, we don't really see a resistance develop, so it's a good tool for there. Medium-risk products, low-risk products, daconil, chlorothanol is really the lowest-risk product we have. There's never been a documented case of resistance in the decades that chlorothanol has been out. So why does rotating and tank mixing help? Right? We talk about, anytime we talk about fungicide resistance management, we always talk about rotating and tank mixing. Well, we're not trying to, keep at bay all resistance. What you're trying to do is keeping resistant isolates at bay and keeping them from dominating the entire population, right? So you don't want one single isolate that is resistant to DMI fungicides to spread and take over that entire population, right? You're never going to eliminate that resistant isolate, but you don't want it to take over and dominate the entire population of dollar spot. So to kind of quickly illustrate that, I have a picture here. This is I took it online from Fossil Trace Golf Club out in Colorado. Nothing against the course. It just had the best overhead view of a putting green that was online. So I'm guessing they don't have much dollar spot out in Golden, Colorado. But if we assume that all of these blue and red spots are dollar spot isolates, the blue ones are sensitive to DMI fungicides or SDHI fungicides, and the red ones have some level of resistance. We go ahead and we make our application. We are drastically decreasing the population of the sensitive isolates, whereas the resistant isolates are inhibited less, and they're faster to come back and recover from that. So they spread, they take over more of the population. Now, if we come back with the same fungicide again, or the same fungicide class, we're going to continue to select for that resistant population over time after we make another application. Now we've got a population that is highly resistant to whatever class we've been focusing on, and is now much more difficult to control. So if we would have rotated, we would have kept these resistant isolates from the different classes at bay for a longer period of time and not allowed it to develop to this problematic state. All right, so if we kind of revisit a similar graph to how this might look from a different perspective, right, this graph we saw before, here's that fungal growth on the left, and now let's just focus on emerald, and let's say we have a resistant population of dollar spot to emerald, right? So we have our same level, dollar spot over 25 leads to symptom development. Now we have a resistant population, so we spray our emerald here. It is inhibited less than what we expect, and once it starts to grow, it can actually grow back, and now it grows back to a level that is greater than symptom than what's required for symptom development. We apply that same chemical again. It's knocked back even less. The fungicide is able to recover faster, and it grows out, and we're seeing breakthrough on products before we would typically see it, right? If we're expecting 28 days, we're seeing dollar spot occur, somewhere around 24, 25, 26, whatever it might be. All right, so one of the main tools then that we always talk about is rotating products or rotating classes. One of the best ways to do that is to use the FRAC codes on the fungicide labels. Bannermax is a group 3 fungicide as labeled by FRAC, the Fungicide Resistance Action Committee. So is Mirage. It's a group 3 fungicide. They're both DMI fungicides. So if you want to make sure you're rotating amongst fungicide classes, just rotate amongst FRAC code numbers. It doesn't do you any good from a resistance management standpoint to rotate between Mirage and Bannermax. They're both in the same class. They're both going to develop resistance in the same way. So another product, Exemplar, it's an SDHI fungicide. All SDHI fungicides are in this group 7. If we go to a mixture, there's a lot of mixture products out there. They're going to give you the FRAC code for both active ingredients, or if there's 3 active ingredients, you'll see 3 FRAC codes listed up here. 3 is for the DMI fungicide. The M is for the chlorothanol. M means multi-site mode of action. So if you have an M on there, you know that you have a multi-site mode of action on there. Normally, it's going to be chlorothanol. So if you're missing, not all labels have FRAC codes. So if you want to look at what a FRAC code is for a particular active ingredient, just go to the FRAC website. There's a website here, and it's included in the handouts as well. And you can very easily and quickly look up the FRAC code.
Paul,
Yes.
Can you go back to that slide for just a second?
Sure.
Because what I have to say here is, remember the label is the law. Our audience, many of them know I say that, but we are very concerned about that. There are times when some of our researchers are using things and they're using them at a different rate. I don't believe that's the case in what Paul's talking about today, but we want to make sure that we always comply with the label, including that it's labeled for golf course use when that's where we're using it. Thank you.
Yep, important point. I'll be teaching our state of Wisconsin applicator training class tomorrow, and we say that same line, the label is a law, about 700 times throughout the day. So
excellent. Hava would be proud.
All right, what about some additional strategies just besides product tank mixing or rotation? Know which of your fungicides are the most susceptible to resistance development and when those diseases are most active. Typically for dollar spot or anthracnose or Pythium root rot, that's going to be during the middle of the summer. So we want to make sure that we're either not applying those sensitive fungicides at that point in time, or we're tank mixing them with other modes of action or with chlorophanol. We can also use fungicide alternatives or plant health promoting products. These are basically kind of like fungicides, but they have a different mode of action. So iron sulfate on dollar spot is basically kind of a fungicide, but it doesn't have the same mode of action. Phosphites or other plant health promoting products, they can limit disease without the same selection pressure on the fungus. And here's the quick picture. We're doing some work. We have a GCSAA funded study looking at iron sulfate and urea to control dollar spot. We're going to start that this upcoming summer in a collaboration with Minnesota. And this is a course that we worked with in Toronto. He accidentally left a check plot. This was a skip on his iron sulfate. You can see how high the pressure is. Iron sulfate over here, iron sulfate over here, no dollar spot, and a pretty good pressure in this area. So those can be useful products in helping to limit the selection pressure on the fungicide or on the fungal population. So we're kind of running out of time here, so I'm going to move through these example programs here pretty quickly. If you have any questions on these programs, feel free to send me an email. This is just a potential program that I might recommend to a superintendent managing dollar spot in the Midwest. We have multiple chemistries, SDHIs, here in the middle of the summer, July 1st, July 21st, we might have our highest dollar spot pressure of the year. We're going to go ahead and we're going to tank mix in chlorophanol to keep to manage that population. You always want to make sure that you're limiting a single class of fungicides to really 2, maybe max 3 applications per year targeting a specific disease. I talked with Bruce Clark over at Rutgers, got an example program for anthracnose, and same thing. It's basically the same thing, but just looking at a different disease. So we're looking at some different products. During the heaviest points of the year, the highest disease pressure areas of times of the year, we have multiple active ingredients mixed in from different classes. We have mixtures with chlorophalanol, and Bruce has a note down here that QOIs and benzimidazoles can be used if you don't have resistance, but I'm guessing there's pretty widespread resistance to those products out in the Northeast. We don't really have a lot of anthracnose resistance in the Midwest. We just don't have the same level of pressure. Talked to Bruce Martin down at Clemson and got a program that he would recommend for managing all the various types of diseases, including Pythium root rot on bentgrass in the Southeast. Obviously, really, really high disease pressure under those really hot, humid, wet conditions in the Southeast. But really, the same principles apply. During our heaviest pressure times of the year, we're mixing multiple classes. We're mixing in contact fungicides when available, depending on the disease that we're looking for. Right, a contact fungicide won't do any good for a Pythium root rot, but it will be helpful for other diseases. So these are some example programs. Feel free to look at these programs and contact either myself, Bruce Clark, or Bruce Martin if you have any questions on these. So just a couple final thoughts as we wrap up here. Resistance management is important, and it's increasing in importance due to some of the things we talked about on that first slide of the presentation. Fewer new products, increasing pressure on the products that are already out there. In my mind, it's difficult, if not impossible, to completely eliminate the development of resistance, but certainly these implementation of best management practices for resistance management is critical in greatly slowing it down, so we extend the life of these products that we have. And other actions like precision disease management, fungicide alternatives, host resistance. There's some really great dollar spot resistance bentgrass cultivars coming out. Those are all important for mitigating the development the development of resistance. So with that, that's all I have. I want to thank again GCSAA for the invite. Thanks to PBI for the sponsorship. And just like you rely on your teams at your courses, this is my team, and I rely on my team very, very heavily to do the research and the extension, the diagnostics. And so I'm very grateful to all the help that I get from them. Feel free to go ahead and email me with any questions you have. Give me a call at any time, and I'd be happy to discuss any questions on fungicide program development, disease diagnostics, precision disease management, any turf-related question that you can think of. Always happy to talk about that with everybody. So with that, I'll go to the last slide and the code points, the points that everybody's really interested in. And with that, I'd be happy to answer any questions if there's any in the queue, Lisa.
Yes. So there's a lot of talk on Twitter about iron sulfate. What form, what rate, etc. would you recommend?
Well, so in my mind, the rates are going to be pretty site specific. We've seen certain rates. Certain superintendents go up near 5-6 ounces and had decent success with not a lot of discoloration. I think most superintendents are down to that 3 to 4 ounce rate of iron sulfate and seen pretty good success with that. I wouldn't consider iron sulfate with it to have the ability to completely eliminate fungicide usage. Typically, what happens is you can keep that fungicide population or the fungal population knocked down a little bit better. You're kind of just slapping it a little bit and keeping the population tamped down a little bit better and increasing the effectiveness of your fungicides. But again, superintendents are all over the place on the rates they're using with iron sulfate.
Okay, great. As you can see, the webinar has been approved for external education points, and so you will want to enter the code 999-20717-27537 in the external education affidavit on GCSA's website. Anyone who might be listening to this as an on-demand in the future, please make sure you use the date that you're listening and not our original date to make sure your points are awarded in the correct renewal cycle. Next question, are there product names for iron sulfate?
There are. I don't know them offhand, unfortunately, but there are different companies have different products that include iron sulfate. So I would talk to your local fertilizer rep to get some of their products. And there's more and more formulations coming out as the popularity of this iron sulfate grows, definitely.
Okay, excellent. If you have other questions, you can raise your hand now or type that in the question and answer box. Next in the series from PBI-Gordon, Hybrid Bermuda Diseases and Control Options on April 17th. You can enroll online for that or any of our other webinars in the regular education series from Syngenta, or the equipment management series we have going on tomorrow. Also, if you might be interested in teaching for GCSAA, hint, hint, Paul, there is a Teach for Us that is open on the website. Next year's GIS is in San Diego, so that's always a good location for everyone to come. Is there much development going on for new classes of fungicides?
Not to my knowledge. I'm sure there is a lot of research. Right? It costs about $300 million and 10 years to bring a fungicide product to market. So I'm sure there's a tremendous amount of research and development going into looking at new classes, but I don't know where those are in the pipeline. I know a lot of the new chemistries that are going to be coming to the market over the next few years are SDHI fungicides, and they'll all likely have some little niche that makes them very effective in a certain manner. But from a resistance management point of view, we do need some additional classes. So hopefully we have something new coming down the pipe soon, a new class coming down the pipe soon.
Okay, and back on the iron sulfite, is that available granular or just spray?
To my knowledge, it's available both, but most people that I talk with spray it.
Okay, and then when combining products, can we use lower rates of each product with the same efficacy?
Yeah, it'll depend on the pressure it'll depend on the products you're using, it'll depend on the pressure you're seeing, so kind of depend on the time of year and depend on the disease. Typically, when you combine products, you will be able to lower those rates. But again, it kind of depends on the products that you're using. You kind of look at it as if you're applying two different products, you're going to have an increased inhibition or an increased dieback of that fungal population. So typically, you can go with lower rates and still see the same level of efficacy. But again, it depends on what you're mixing, the disease pressure. If you have really high disease pressure, I wouldn't drop rates a lot, but it kind of depends on a lot of different things. But yeah, normally you can.
Okay, I'm not seeing other questions at this time. Paul, do you have any closing remarks for us?
Just best wishes on a great season. I'm looking forward to getting out there in the field and interacting with superintendents all over the country and over the course of the upcoming season. So best of luck, everyone.
Thanks for teaching again, Paul. Appreciate it. Everyone, thanks for joining us. There will be an evaluation survey as you exit today. If you would fill that out, we would sure appreciate it. And thanks, everybody, for joining us. Have a great day.